Thank you again. As Phil mentioned, our work is really centered around this three big systems in our biological system. Immune system is really our key focus here. And as you heard, we think that the immune system is perturbed during ME/CFS, but the question is how. And whether the microbiome and related metabolomics, metabolites either from the microbes or by the host is one of the reasons for this changes. Because if we were to identify these, not only would they be biomarkers, but as Ian just mentioned, they can be actionable targets for therapeutic intervention. So unfortunately, immune system is very complicated. You have different compartments of immune system that protects us against viruses and various pathogens. You have innate cells and B cells and T cells, and each of them have many subsets. And my lab has been working on T cells in particular, because these are sort of like the commanders or the regulators of the immune system. And there's a huge labor of division within the human T cell populations. They develop in response to different threats, bacteria, viruses, parasites, and so on. You even have cells that can attack tumor cells, and they're cytotoxic. You have invariant cells, non-classical cells that don't require peptides, but recognize some other molecules, as I will mention in a minute. But they're also sort of double-edged sword, because you can have autoimmunity if their function is not correctly controlled. And we even have a subset of cells called Tregs, regulatory T cells, that function to actually control the rest of the T cell population, so that to make sure that they don't go over the board and they don't cause too much trouble, and really focus on the infection. And in fact, there are even more flavors of these T cell subsets. Within the subsets, you have subsets. And this complexity is derived because you have different types of cytokines and chemokines. These are small molecules that send commands to different cell types produced by different subsets, but they also have to be localized to certain regions. For example, certain cells like to go to gut, others to the lungs, others to liver, and so on and so forth. So you have this compartmentalization in subpopulations that we're still trying to define all of these subpopulations. In the depth of the observation, both in terms of frequencies and in terms of functionality. So for this study, we've been working with Bateman Center. We've obtained 150 patients, recruited them through Cindy Bateman's group. And these, we've acquired 3 time points, blood and fecal samples from these patients. And these were divided into 2 cohorts of less than 4 years and more than 10 years of disease. Turns out this is really important to divide the patients like that. And we've analyzed we are analyzing their immune cells and functionality, and we generate about 300 immune cell and function data points per patient, as well as the microbiome and metabolomics data that I will show you. We think that this will generate maybe one of the largest prospective immune profiling data in depth and breadth in ME/CFS patients. We've also now included about 40 long COVID patients that will be followed at least 3 time points, and we hope to compare those data both immunologically and metabolic in terms of metabolism with the ME/CFS patients. In fact, in a previous study, we've already began to identify major differences in ME/CFS in healthy patients in terms of their immune cell functionality and frequencies. I won't go into details here, but we'll mention 2 of them in a minute, which turn out to be related to the microbiome perturbances, as at least we think so. We've also performed and will be performing a machine learning analysis to these immune profiling data sets. And the goal here is to both identify sort of biomarker sets. In fact, using our previous data set, which was a cross-sectional data a single time point, we were able to identify who's a patient and who's a healthy control around 90% of the cases by just looking at their immune profiling. So this was very, very encouraging for us. We think that we can even get better at this by incorporating other data sets. The other important point is you heard from Ian and before from Nat that the microbes within us turn out to be really important. And these microbiome, as we call it, is also personalized. It can be different from person 1 to 2, 3, or 4, and you have thousands of different strains of bacteria species that some of which need to be still identified. So this is a very laborious, but we think a very important side of the work. And furthermore, this gut microbiome community is dynamic and it can change by disease process, by diet, antibiotics, pathogens, immunodeficiency, and so on. So it's important to look at it over time, or as we have done in different cohorts, less than 4 years of disease and more than 10 years of disease, to see what happens. There's a very fine equilibrium in this microbial ecosystem, because not only you have these symbionts or commensals which help us digest food, and they produce lots of metabolites that shape our immune system and other biological functions, but you also have these bacteria that are called pathobionts that can actually cause trouble if this balance is perturbed, and they will cause inflammation or called dysbiosis, which of course through effects on the immune system will have a wide range of chronic problems in our body. So our microbiome project design was to get fecal samples from stool samples from these patients that I just mentioned, which we sequence. We also look at the plasma metabolites, as I will mention in a second. And the idea is to not only look at these microbiome on its own, but also try to link them to bacterial metabolites and of course, eventually to the immune system. So we have some early findings. We're still working on the very, very large data sets. We see major microbial dysbiosis, but specifically in patients who had disease less than 4 years. Those who had more than 10 years, they start to reestablish and kind of look like healthy controls, but still they have major metabolic dysbiosis, some sort of a major metabolic dysfunction that suggests an inflammation. It's not clear why, whether life changes or GI disturbances or microbial changes lead to this long-term dysbiosis. But again, this shows that the value of these two cohorts in this sort of multi-omics analysis, because again, as Ian mentioned, this is a very heterogeneous disease, and it's really important that we compartmentalize the patients. We have other findings which look quite similar to what Ian described, actually. So this is very satisfying to see reproducibility by different centers and different cohorts looking at same type of subjects. And so again, our framework of goal here is to understand the microbiome, but just looking at the microbes per se may not tell you the whole story. We really need to understand the metabolites that they produce or by the host or by the bacteria, and how does that link to the physiology and immunology. And of course, we want to correlate that to the host phenotype. As I mentioned, the duration of disease seems to be a very major factor in this process, and we are sure that there are other demographic and other clinical features that will further stratify these patients based on their microbiome, metabolism, and immunological data sets. One of the things that we actually found in our previous study is that a key cell type called Th17 cell that is shaped by the microbiome, and this is very well established by certain bacteria, is involved in a variety of autoimmune diseases, is also perturbed in ME/CFS patients. This was one of the major populations of T cells that we find to be disrupted in ME/CFS patients that we hope to reproduce with the new cohorts as well. So that was our first link to the microbiome and the immune system. And the second is that we've also been working on a non-classical T cell type called mucosa-associated invariant T cells, which actually recognize bacterial metabolites derived from bacteria-produced riboflavin or vitamin B2. And they seem to be sort of the guardians of the bacterial ecosystem in human body, particularly in gut and skin. We don't exactly know how they function there, but we think that they really sense the changes in the microbiota, and they can either get activated or they can get suppressed. And we see, again, a major overactivation of these mate cells in ME/CFS patients, suggesting a significant perturbance of the microbial system. So these were the two of the major links to the immune system and the microbiota. And finally, metabolomics analysis, we think, and in fact, turn out to be very important. And the idea here is, again, to perform a targeted metabolite analysis in blood sample. We've chose a company called Metabolome. They have a selection of up to 1,000 metabolites. And we perform this again in multiple time points, 3 time points that we take the samples from patients 1 year apart. And so far, we've accumulated over 500,000 data points. You can imagine this is really a massive data that we're still mining right now. And again, the key goal here is to link metabolism to the immune and the microbiome perturbation. So some of these metabolites are produced by the microbes, and whether they actually also link to the immune dysfunction. Of course, they might link to the other dysfunctions, for example, neurological dysfunctions as well. And in fact, we've already began to find bacteria-associated metabolites such as short-chain fatty acids. Jan mentioned butyrate. In fact, we also find that these bacteria are reduced secondary bile acids, tryptophan pathway metabolites, all of which act on the immune system and produced by the gut microbiome are perturbed, appear to be perturbed in patients. And in fact, we have also one last assay that we want to identify specific metabolites produced by bacteria that are not known yet to be identified. So I won't go into details. We have a very detailed flow assay for this. Again, I might be breaking up a little bit. And the idea here is to screen thousands of bacteria derived from patients and find new metabolites. And we already found some metabolites that activate a very important pathway called aryl hydrocarbon receptor pathway, or AHR pathway. And we're quite excited about that. Again, this is a very actionable type of work that could lead to potential therapeutics. Just to summarize that we are now at the final year of the of our grant and center, and we are trying to integrate all of these massive data sets and identify new hypotheses, new potential mechanisms that can identify a certain subsets of patients or certain mechanisms that we can either use as biomarkers or, again, as Ian mentioned in the last slide, as potential targets for clinical trials, because we think that metabolites and microbiome, again, are probably the low-hanging fruit for such clinical trials. And it will be very interesting to see how that moves forward. Lastly, as I mentioned, we also have a cohort of long COVID patients that we think could be informative, because we don't think everything is the same in long COVID as ME/CFS, but there are some similarities. And in fact, this might be the very early stages of an ME/CFS patient. So it's also a unique opportunity to understand what sort of changes happen in the immune system and metabolism in these subjects. So I'm going to stop there. Really thank our clinical collaborators, Suzanne and Cindy. We couldn't do any of this without them. Julia O is our microbiome lead. She's done most of the microbiome work. Zudang Yao identifies bacterial metabolites. Shuzhao Li is our metabolomics lead. He's doing all the analysis, and Duigo is our computational biologist. Will be working on a lot of data analysis. So I'm going to stop there. Sorry for the interruption.
Thank you, Daria. So just a couple of
Happy
questions.
to take any questions.
Yeah, thank you, Daria. So I thought the nervous system was complicated. I think the immune system's 10 times more complicated. So one of the questions that came in was, you've looked at the difference in age and length of illness. Have you looked at your data, the differences between males and females?
Yes, in fact, age is a major factor. Both 50 and under 50. In healthy controls, we see big differences in immune system as well as the ME/CFS patients. Men and women are definitely also different in their immune profiles. We didn't have a lot of males, unfortunately. 85% of our patients were females, so we may not have statistical power. But Duygu Uçar in our center, she's been looking at the elderly females and males, and she finds huge differences in the male and female immune system. So there's no question that they're widely different, and that's a very, very important point.
Yeah, and I think some of the most important or interesting data that you're showing is the difference between those with shorter duration versus longer duration illness. What do you think is happening that is that difference? Are the individuals with longer duration illness compensating in some way, or what are your theories there?
Yeah, that's a great question. That's certainly a possibility. For example, we see the same thing in the immune profiles as well as the microbiome. In immune profiles, patients over the age of 50 start to look again more like the healthy controls, because the healthy control immune system is also changing. There's an aging of the immune system itself. But the young patients, because we, of course, compare them to the age-sex match, the controls, they kind of look more accelerated aging of their immune system, as if they're moving faster, or there's more a disturbance in their blood. So we see more changes in those patients, how that relates to the disease duration per se. I mean, age is a factor, but probably many other things are a factor, and the microbiome seems to start to look like the healthy controls. I don't have a good answer, but what you suggested is definitely a possibility.
Yeah, thank you. And just if you could just sort of reiterate what the mate cells are, because those are, I think people might not be as familiar with mate cells, what their role is.
Yeah, mate cells are very interesting. So they're called non-classical T cells because normal T cells recognize peptides derived from any type of a protein that's presented by what's called an MHC molecule, whereas mate cells recognize small molecules, metabolites, from one that's known is from a riboflavin pathway. Riboflavin is vitamin B2, which is produced by bacteria. As bacteria is producing this vitamin, it goes through different stages of the metabolism, and it produces certain metabolite byproducts. And one of those actually bind to mate cell receptor and activates it. We think there are other ones like that. So they really are sensing those bacteria that are producing this metabolite very directly. And they're actually very large in population. They're not few. Some people have up to 30% of their subset of T cells formed by these cells. Others have only 1%. And there's such a huge variation in numbers. We think that could be a reflection of their microbiome. If those that have very active microbiota maybe have more mate cells, that they need more mate cells. But it's a very interesting, they're cytotoxic. They can kill intracellular bacteria like Salmonella, for example. So that's another of their function. They seem to play a role in COVID actually, and they go into the lungs. It's a very interesting subpopulation that we still need to learn more.
Great, thank you very much, Daria.